guys. This is the first video of hopefully a 10-part series of me reviewing AP Biology for you guys. I'll try to post every week, maybe twice, but I don't know if that'll happen because I have other things to do. But today's video is on cellular respiration, and my next video is going to be on photosynthesis, so just keep sending requests for stuff, and let's get to the learning. Learn! Alrighty, so how this is going to work, I have like what we're going to go over here, and if you just follow the arrow, that's where we'll be. So first we have the definition. So like, what is cellular respiration? It is the process by which organic molecules are broken down in order to produce energy in the form of ATP. So like, just like it's, if you scale it down, this is the cell's way of like eating. So it takes energy out of glucose, out of proteins, out of fats, out of all that jazz. And that's just kind of the process that we do it, and we got to go over it in depth because AP wants us to. So that's that. And next we're on to redox reactions. Alrighty, now on to redox reactions. So you might be wondering, why does this breaking down of organic materials make energy? And that's because in the reaction, electrons are moving. And electrons you can kind of think of as energy. So like electron, electricity, electricity is energy, electrons are energy. So when an electron moves from reactant to reactant, that's a reduction-oxidation reaction shortened to redox reaction. So what we have in cellular respiration is a redox reaction. So something you can remember to deduce whether it's reduction or oxidation is this little Leo goes grr. So Leo is like a lion. Yeah, and grr is the sound a lion makes, so like grr, RxD, yeah, all that jazz. So Leo, if it loses electrons, it's oxidation, and for grr, if it gains electrons, it's reduction. So that'll help you guys to figure out which is which. But here's the actual formula, and shows what's oxidized and what's reduced. So the glucose, the C6H12O6, is oxidized when it goes to 6CO2, but 6O2 is reduced when it goes to 6H2O. So that movement of electrons creates this energy here, but you might ask, how are we moving the electrons? That's when we get into the fancy-dancy electron carriers. Alright, so like, electron receptors, they're freaking cool. There it was. So yeah. They're pretty cool. But here's why they're cool. So there's two electron receptors used. The most common one and the most versatile one is NAD+, which goes to NADH when it's reduced, and it creates 2.5 ATP once it goes to the little motor thing in oxidative phosphorylation. But then we have FAD, which goes to FADH2, which makes 1.5 ATP during oxidative phosphorylation. So what makes them so cool is that instead of going straight from glucose to energy, which would be like an explosion, it does it in steps, so we can harness the energy from each of those steps and make ATP from it instead of just burning yourself from the inside, which sometimes is nice. But they're also pretty cool because they go in between these two states fairly easily. So they can be reduced pretty easily, but you can also oxidize them pretty easily, which means they can lose their electrons and gain electrons really easily, which makes all these things where they are oxidized and reduced so often so nice. So yes, that makes them pretty awesome. And now we're going on to aerobic respiration. So now, into an overview of aerobic respiration. So aerobic means with oxygen, so it uses oxygen in the process, so it works good. It generates 30 to 32 ATP depending on how much you got to do to get it done. So sometimes you have to spend a little more energy, so that's why you have a lower ATP. And it's composed of the 4 steps of glycolysis, pyruvate oxidation, citric acid cycle, and the electron transport chain, also known as oxidative phosphorylation. And this is what we do because we can breathe, and that's kind of why we have to breathe, so we can get oxygen. So now, let's go into the diagram for how much ATP each step produces. So here's kind of a rough diagram of what happens during aerobic respiration. It starts with the breakdown of alrighty, so here's kind of a rough diagram of what happens during aerobic respiration. Starts in the cytosol, or the cytoplasm, where glucose is split into two pyruvates and it creates 2 ATP. Then the pyruvates are chucked into pyruvate oxidation, which produces 2 NADH in the mitochondria. Then it goes to the citric acid cycle with acetyl-CoA, and that produces 2 ATP, and 6 NADH and 2 FADH, and then all of those electron receptors go into the electron transport chain, which produces 26 to 28 ATP depending. And this is just for glucose. Every single different organic molecule produces, it comes in different steps. And that's what I'm actually going to go over next. So, this is where different organic materials go into aerobic respiration. So carbs are just like sugars, so like glucose, they're just the same. They just go straight down the line like we're used to, produce the regular amount of ATP. So proteins, they enter as amino acids, individual amino acids, and like you can get them from meats and stuff. Just when something says protein, this is what you're eating, it can go in as pyruvate, but it has to chuck off an NH3 group. Or it can go in at an acetyl-CoA stage or at the citric acid stage. It really depends on which amino acid it is, because they're all different. And it'll produce different amounts of ATP, because sometimes it'll go through pyruvate oxidation a bunch of times, or it'll go through the citric acid cycle a bunch of times. So it can make a lot of ATP that way. And it's the same thing with fats. Glycerol can go in at this stage of glycolysis, which is when it is glyceraldehyde with a phosphate group attached. So it can go into that, and it can be cut lots of times because fats are big, long hydrocarbons. So they can just be chopped up, and they can fuel ETC a lot. It can make a lot of ATP. And with fatty acids, they enter at the acetyl-CoA stage, and they can go through the citric acid cycle, and the same thing, they can make a lot of ATP. It really just depends, but the main thing we focus on is the carbs with glucose, just going straight in, because that's like our most common way of getting energy. So now, we're going on to glycolysis. So, glycolysis is kind of cool, because it has 3 phases. It has the energy investment phase, where 2 phosphates from ATP are inserted onto glucose, because phosphate has a way of making molecules much more reactive, so it can actually do stuff with glucose. So that's where 2 ATP are invested. But then after glucose has been ripped apart, something called substrate level phosphorylation happens, where it's literally just the synthesis of ATP from ADP. It's not forced together like oxidative phosphorylation. So that forms 4 ATP. So 4 minus 2 is 2. So you get 2 net ATP produced. But that's not the only thing that happens during glycolysis, and it's vastly more important stuff happens. So glucose and 2 ATP are what it takes to do this, but it produces 2 pyruvate, which are the molecules that like make all this stuff happen. So they're used a lot further down in the cycle. It produces the 2 ATP, which is nice, 2 NADH, which are used for the electron carriers in the electron transport chain, and 2 H2O is produced. Something that happens is no CO2 is produced by this, because glucose is a 6 carbon, and each pyruvate is 3 carbons, so no carbon is lost during this. And glycolysis is also pretty interesting, because it's been found in like really old like archaeobacteria. So if on the test they ask how is glycolysis an example of evolution, and it's because glycolysis has been found in organisms a while ago, and we use this mechanism pretty much the same as they did. So that's cool. But now we got to deal with this dude right here, 2-pyruvate, in pyruvate oxidation. Alright, now for pyruvate oxidation. This is like the shortest and the simplest step, and it just happens from when the molecules of pyruvate enter the mitochondria from the cytoplasm. So this process happens twice. The first thing that happens is this is a pyruvate molecule. It releases a CO2, so it kind of just breaks this bond, and this little CO2 floats off, and it's released. So that's one of the 6 CO2s released. And then this molecule is called acetate, but what happens is it gets oxidized, and electrons are ripped off of this acetate, and it charges NADH. So it's like our batteries with the electron carriers. So that goes down the line. And then the third step of pyruvate oxidation is the addition of coenzyme A from vitamin B. So we have to kind of just, so this is gone, and we add a CoA, and now this is acetyl-CoA. And that's what we use to go into the citric acid cycle. So let's get on to that. Alrighty guys, now for what I think is one of the most complicated parts of this, the citric acid cycle. So it's called the citric acid cycle because the starting molecule is citrate, and then the second is isocitrate, but in my head it's just a whole bunch of citrate. But yeah, so it's, all these are produced by just the changing of molecules. There's 8 different molecules in the citric acid cycle that just keep cycling, and they're oxidized, which create the NADHs, and they can be reduced. There's a bunch of different additions that happen, so I'm just going to go through it as we go. So first we start with the acetyl-CoA we got from pyruvate oxidation. So the acetyl group from acetyl-CoA goes in to the cycle, and CoA is kind of thrown out into the cytoplasm to be used later. Then with the change from citrate to isocitrate, there's an oxidation of those, so that loses electrons. Then the NADH is reduced when it gains the electrons, so it's kind of charged now. Then a carbon is thrown off of this, so it went from a 6 carbon to a 5 carbon. And then that happens again, carbon is thrown out, and there's another oxidation reduction, so we charge another NADH. Then the coenzyme A group comes back into the equation, and it's added back in. And during that step, an inorganic phosphate is added to GDP, which then makes it form GTP, and through substrate level phosphorylation, the ADP is charged by the GTP. So it kind of rips the GTP phosphate off and puts it on to ADP to make ATP. So that's how we produce energy from the citric acid cycle. And then the coenzyme A is taken back off the molecule. Then there's the redox that charges the FADH2 that we get from the citric acid cycle. Then a water is put on just to make it more reactive and add some hydrogens and oxygens. And then another redox reaction happens, and it goes again. And because we have 2 acetyl-CoA molecules from the 2 pyruvates and pyruvate oxidation, the citric acid cycle happens twice. So you can kind of count them in this order. So we got 1 NADH, 2 NADH, 3 NADH, and times 2, it'd be 6 NADH from the citric acid cycle. And we get 1 CO2, 2 CO2, so 2 times 2 is 4. Then we also had 2 CO2s formed in pyruvate oxidation, so that's the 6 we need. Then we formed 1 ATP, but times 2, so there's 2 ATP from citric acid cycle, 1 FADH2, but times 2, 2 FADH2s, and that should be everything produced. And now we get on to the actual energy-making part, the electron transport chain, where we take all these NADHs and FADHs and have fun with them. So let's get to it. So now for all this jazz, this crazy electron transport chain and chemiosmosis. So first, we take our big players, the electron carriers, NADH and FADH2. They pretty much power all of this. So the first thing they do is they donate their all of their electrons and send it up the electron transport chain, where it just kind of releases its energy every single time until they have no free energy anymore, really. And they also can pump some of their H+ ions up there, but that's not really where it comes from. But what happens is, so this powers proton pumps. So these all pump H+ ions, which are just protons, up and to form a positive proton gradient, so that positive always wants to go to negative. So the electron transport chain is the first part of oxidative phosphorylation. So next we have chemiosmosis. So osmosis is like, we've gone over this, like water wants to go to where solute is. So this is kind of like osmosis but with chemicals. So the plus wants to go where the minus is. So because there's such a strong gradient, it wants to go to its opposite. So it goes through this little funnel in a protein called an enzyme called ATP synthase, which works a lot like, think of like a water mill. Like water forces it to do work. The H plus is the water, the ATP synthase is the water mill. So with the H plus going down, it powers a rotor, which then squeezes an ADP and a inorganic phosphate together to make an ATP. So it kind of works like a machine. So it squeezes the phosphate on, and it forms all the ATPs from that. And then when you're done, you have a bunch of H plus ions all down here, and you have the oxygen, which now is where the oxygen comes in. So then you also have the electrons coming down from the electron transport chain. So all of those combine to make a stable water molecule, and that's where we get all the water from. So it's a very cool, ingenious plan, and I think it's cool how this all happened, like with the evolution and such. This all can happen though because of the mitochondria and its membranes, within the cristae, so that it can actually separate the H plus from the negative. So that's quite interesting, and it's good that another mechanism of this is because we recover our oxidized forms of NAD plus and FAD. That means they can go back and be used again. So it's it's a cycle. The cellular respiration is a cyclical thing, and everything we use, everything we made, we can use again. So that's quite interesting. But there are a few differences between aerobic respiration and anaerobic respiration, which comes next.
Morty, I turned myself into an anaerobic bacteria.
Yeah. But anyway, anaerobic respiration. So it's just aerobic respiration, but in the absence of air. Well, like aero, but it's the absence of oxygen. So why does oxygen matter? As you saw in the electron transport chain, oxygen acts as an electron receptor because it's so electronegative, which means it just like attracts electrons. So it kind of rips the electrons down through the electron transport chain to take them. But without that, stuff doesn't work right, so it halts after glycolysis. So it's a very simple thing. It's found in like some of the oldest organisms, and it's a very simple way to make energy. So just like aerobic respiration, it starts with glycolysis, but then it can't go any further. But why does it need to do fermentation? It's because in glycolysis, you remember that we form NADH, but because it can't go any further, it can't take NADH back to NAD+, to be formed back into NADH. So you just end up with a huge stockpile of NADH without doing something to change it back to NAD+. So that's what fermentation does. It changes the NADH back to NAD+, and it has to make some other stuff with the pyruvate. So we'll get into that. But as you can see, the diagram for anaerobic respiration is significantly simpler. So you start with glycolysis, where you have 2 NAD plus and 2 ADP, and you get the 2 ATP through substrate level phosphorylation, and 2 NADH, and you get the 2 pyruvates from 1 glucose. Then you go into fermentation, where you put in 2 NADH and your 2 pyruvates, I forgot those, and you get out 2 NAD plus to be used again, and 2 lactates or 2 ethanols, depending on which type of fermentation you use. So it's a nice little cycle, and it feeds itself, and it works for bacteria pretty well, and it even works for humans, but we do lactic acid fermentation when we don't have enough oxygen to perform aerobic respiration. So that's why when you do like really heavy lifts, your muscles will be sore, because since you need to produce so much energy so quickly and with little oxygen, it has to just produce as much energy as possible. So it has to resort to lactic acid fermentation to produce energy. So there's lactic acid buildup, which hurts your muscles. So that's a kind of a cool little fact there. But now on to how glycolysis works. This is probably the shortest clip because glycolysis works exactly the same in anaerobic respiration. I'm gonna start this over so I can. Yeah, good. This is probably going to be the shortest clip, but anaerobic respiration is glycolysis, and aerobic respiration is glycolysis, exactly the same. Because it's the same mechanism, and that's because of evolution, BB. Alrighty, so this is lactic acid fermentation, and it's slightly simpler than alcoholic fermentation. So, like, that's why a lot of simpler things use it, but it's just one less step. So you take the pyruvate, which is this molecule, the O, it'll be C3, it's a 3-carbon little thingy, and with it acts as the electron acceptor to NADH. So it accepts the electrons and the H ion, well the H's, and it forms 2-lactate, which is this molecule, and it is the ionized form of lactic acid. So it's a pretty simple. Just you can see they're very similar. It's just the addition of an H. And because of that H, it takes off one of the bonds in the double bond, and it's a very unique way to do it. But now on to alcoholic fermentation. Alrighty, now for the second form of fermentation, alcoholic fermentation. So this is slightly more advanced because it has a second step involved. So there's actually 3 molecules, so it goes from pyruvate to acetaldehyde, then to ethanol, or which is drinking alcohol. So it starts with the pyruvate and the 2-NADH, and this pyruvate releases this part of the molecule, which is just the same as a CO2, so that is released into the air, and there's 2 of them because it's 1 for each pyruvate. So that forms acetaldehyde, which then, shifted over again, is to ethanol with the addition of these hydrogens. So then we have the 2 molecules of ethanol and 2 molecules of CO2. So alcoholic fermentation produces both ethanol and CO2, so it can actually kind of carbonate it while it adds alcohol. So that happens with a lot of drinks. So, and then the H is taken from that, and the electrons accepted from the NAD+, make it so that it is, from the NADH, make it into NAD+, which means it can be used again. And we actually have harnessed alcoholic fermentation for a lot of things. And we actually have harnessed lactic acid fermentation for like making cheeses and stuff. So leave it to humans to take advantage of biological processes. But that's pretty much it. It's a pretty simple process, and we'll get to the closing. Hi guys, I just wanted to take this time to thank you all for watching. I'll try to get this out as soon as possible. Another one as soon as possible. I'm going to try to do photosynthesis, but please leave comments and stuff, and smash that like button. It is a lot of work, but it's not too bad. I'll enjoy it, and hopefully all the videos after this one will go a little quicker because I have like a structure, and yeah. So send any requests in the comments, or if you want to message me another way, you guys know how. And that's pretty much it. So thank you guys, and right now I don't have Wi-Fi because our Wi-Fi is down, but I'll try to get this up as soon as possible. So see you guys soon.